Welcome to this explainer. You know, have you ever actually stopped to wonder about the unseen, behind-the-scenes laboratory magic that ensures our medical and scientific tests are flawlessly reproducible every single time? Well, today, we are decoding exactly how scientists tame raw biological materials. We're going to focus entirely on how laboratory antigens reach standardization, because honestly, this is the foundational process that makes accurate medical diagnoses even possible.
Okay, let's dive into this. That exact question, how to guarantee a test works perfectly every time, is the driving force behind the meticulous process we're exploring today. Just think about it. If you take a blood sample from a human or even an animal, you need absolute certainty that the resulting diagnostic test is accurate, right? Well, the only way to guarantee that is by perfectly standardizing the biological materials used in those tests, specifically the antigens.
So here is our roadmap for today. We're going to cover the antigen blueprint, explore the Goldilocks principle, look at 3 antigen workhorses, walk down the path to standardization, and finish up at the quality control checkpoint. Let's get into it. Step 1, the antigen blueprint. Before we can standardize anything, we have to know our raw materials. Now, what's really interesting about this is the critical distinction we need to make right out of the gate. The difference between an antigen and an immunogen. It's kind of like, think of an antigen as a keyhole, and an immunogen as a key that actually turns and starts the engine, meaning it actually induces an immune response. While all immunogens have that keyhole, not all keyholes will start the car. Because not all antigens are naturally capable of inducing that response on their own, careful laboratory preparation is absolutely crucial. We literally have to prime these substances to work perfectly in our diagnostic tests. Moving right along to part 2, the Goldilocks principle. This is where we get into the exact dose and concentration. To understand why concentration matters so much, we need to look at the antigen-antibody lattice. I want you to imagine building a bridge out of microscopic scaffolding. For tests like agglutination or precipitation to actually be visible to the human eye, we need a perfectly balanced bridge between the antigens and the antibodies. When they exist in optimal proportions, they cross-link. They lock together and form massive visible networks. In the lab, the structural sweet spot is known as the zone of equivalence. But look what happens when we lose that balance. It's a classic case of too much of a good thing. In the prozone effect, an excess of antibodies actually interferes with the lattice formation. The bridge just can't connect. Conversely, in the postzone effect, an excess of antigens does the exact same thing. In either of these failure scenarios, the result is a weak reaction, reduced clarity, or, worst of all, a dangerous false negative. This absolutely proves that standardizing the exact concentration of your antigen isn't just a best practice, it's an absolute necessity. All right, part 3, 3 antigen workhorses. Let's look at the tools of the trade. Diagnostic laboratories rely heavily on 3 primary biological tools every single day. First, HA antigens. These are viral or particulate preparations, things like influenza or Newcastle disease virus, and they're used primarily for hemagglutination tests. Second, bacterial antigens. These can be particulate whole cells or soluble components, used a ton in agglutination and serology. And finally, RBC or red blood cell antigens. These are whole erythrocytes carrying surface antigens, and they're vital for passive assays. And this brilliantly illustrates how each of these distinct biological tools, viruses, bacteria, red blood cells, requires its own completely unique validated handling. Which brings us to part 4, the path to standardization. This is where theory becomes hands-on laboratory practice. Let's walk through the red blood cell workflow step by step. It kicks off with an approved blood sample and a quick dose of anticoagulant so it doesn't clot. The cellular fraction is then carefully separated from the plasma, usually with a centrifuge. But the real magic happens next, washing the cells with an isotonic solution before finally preparing the standardized suspension based on whatever that specific test requires. So, the crucial point is why that washing step matters so much. Blood isn't just red blood cells, right? It's totally full of plasma containing free antibodies, fibrinogen, complement components, the whole shebang. If a scientist skipped the washing step, these residual proteins would cause non-specific reactions and literally ruin the diagnostic test. The washing process guarantees a clean, strictly standardized cellular antigenic surface. Now, for bacterial antigens, the workflow shifts quite a bit. After verifying the exact identity of the bacterial strain, the scientist has to standardize its concentration. Because bacteria are microscopic particles, laboratories rely heavily on physical metrics here. They use validated turbidity standards, which is essentially measuring how cloudy the liquid is, or they use spectrophotometric measurements to ensure the suspension has the exact right density. Once inactivated, this precisely measured work in antigen ensures reproducible bacterial clumping every single time it meets a specific antibody. And finally, the HA antigen standardization workflow. This one is a highly precise, sequential mixing process that demands incredible attention to detail. The scientist takes the viral antigen stock and performs very careful serial dilutions in a sterile microtiter plate. Then they add an equal, standardized volume of the washed red blood cells we talked about just a minute ago. After letting the reaction incubate under strictly validated conditions, they read the wells. When they are reading those wells, here is exactly what the scientist is looking for. In a positive HA reaction, the virus successfully attaches to multiple red blood cells, cross-linking them so they stay dispersed. This forms a highly visible diffuse sheet, or a mat, across the bottom of the well. Contrast that with a negative HA reaction, where there's no cross-linking at all. The non-agglutinated red blood cells just roll down the smooth sides of the well and settle to form a tight, compact little button. When standardizing this HA antigen, scientists need to find its HA titer. This is the measure of its hemaggglutinating activity. Basically, they're looking for the highest dilution of the antigen that still produces that diffuse mat we just talked about. Let's say the last positive dilution was 1:64. The HA titer is simply the reciprocal of that number, so flip it, and your titer is 64 HA units. This specific number proves the exact activity level of the antigen preparation before it ever hits a diagnostic clinic. Which leads us to our final step. Part 5, the quality control checkpoint, the ultimate safety net. Just look at how easily standard operating procedures can fail in the lab. It can be a real cascade of disasters if you aren't careful. Got an off-balance concentration? You get weak reactions. Use the wrong species of RBC for a specific virus? The test completely fails. Leftover plasma from a poor wash? Immediate interference. Even a simple, seemingly tiny mistake, like an unnecessary freestyle cycle, can cause total antigen degradation. Let's move on and see how this builds our safety net. To catch all of those potential errors, every single immunological assay relies on 3 essential controls. The positive control proves the test actually can work by showing known reactivity. The negative control proves it won't trigger falsely. And the reagent control acts as a baseline to ensure the diluents and liquids themselves aren't causing non-specific clumping. Running these simultaneously guarantees the absolute integrity of our standardized antigens. So we've seen how raw viruses, bacteria, and blood cells are meticulously washed, measured, diluted, and controlled to achieve perfect standardization. Without this invisible pipeline of preparation, diagnostic immunology literally simply wouldn't exist. So I want to leave you with this. The next time you or a pet go to a clinic and get a rapid test result, ask yourself, how many unseen scientific checkpoints did that tiny drop of liquid have to pass through just to give you an accurate answer? Thanks so much for joining this explainer.